Yeah, please
Is it
start.
ready?
Please start.
Okay, sir. Okay, then I'll
ठीक है.
start. Our distinguished speaker, Professor Anurag Agrawal, Director, Institute of Genomics and Integrative Biology, IGIB, New Delhi. Dr. Shahid Jamil, Director, Triveni School of Biosciences, Sonipat, Haryana. Professor Wasim Ahmed, Dean, Faculty of Life Sciences, AMU. And Professor Tabish, Chairman, Department of Biochemistry, AMU. Professor Riaz Mahmood, dear faculty members, participants, and students. Good morning and Assalamu alaikum. I, Dr. Mohammad Sajid Khan, Associate Professor, welcome you all on the behalf of Department of Biochemistry and Faculty of Life Sciences AMU for the Professor Abdul-Majid Siddiqi Memorial Lecture. The title of today's lecture is The Value of Interdisciplinary Research, a Story of Maths, Biology and Medicine. This lecture was scheduled in March 2020 as a seminar, but due to COVID-19, it was postponed indefinitely. However, Professor Tabish, Chairperson, Department of Biochemistry, managed to conduct it today in webinar mode. Now, before moving towards the today's talk, I would like to pronounce few words about eminent Professor late Abdul-Majid Siddiqi, who established the Department of Biochemistry at AMU, Aligarh in 1986, and was the first chairman of the department. He was also the first dean, Faculty of Life Sciences at AMU. Professor Majid Siddiqi completed his PhD from University of California, San Francisco, after finishing his M.Sc from AMU only. His research was on lipid metabolism, protein chemistry, and enzymology. His research led to the identification of HMG as an inhibitor of cholesterol biosynthesis, which earned a US patent entitled Process of Combating Hypercholesterolemia in 1971. In over 35 years at AMU, Professor Siddiqui supervised 40 PhD and 30 MPhil students and taught thousands of BSc and MSc students. And this memorial lecture is set up by his student and family. Now, before moving ahead, as per the tradition of our university, I would like to invite Mr. Fawad for recitation of Surah from Holy Quran. Mr. Fawad, please.
بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. اقرأ باسم ربك الذي خلق. خلق الإنسان من علق. اقرأ وربك الأكرم الذي علم بالقلم علم الإنسان ما لم يعلم صدق الله العظيم.
Thank you, Mr. Fawad, for such a wonderful recitation and beautiful voice and Kiran. Now, I would like to invite Professor Wasim Ahmed, Dean, Faculty of Life Sciences, who is very good academician and researcher with wide experience in different fields, to say a few words about today's lecture. Please, sir.
Thank you, Professor Sajid. Professor Mohammad Tabish, Chairperson, Department of Biochemistry. Our guest, Professor Anurag Agrawal, Director, Institute of Genomics and Integrative Biology, New Delhi. Our own Dr. Shahid Jamil, Director, Trivedi School of Biosciences, Prof.A. University. Faculty members of the Department of Chemistry, my dear students and participants from around the world. Assalamu alaikum and a very good morning, or depending upon where you are based, good evening or good afternoon. On behalf of the Faculty of Life Science and on my own behalf, I welcome you all to Professor Abdul-Majid Siddiqi Memorial Lecture organized by the Department of Biochemistry, Aligarh Muslim University. We would have loved to invite our honorable speaker to visit Aligarh University, but unfortunately, because of COVID-19 pandemic, our interaction is virtual. For our guest, it is my duty to give a brief idea about Aligarh Muslim University as well as our Faculty of Life Sciences. Aligarh Muslim University is ranked among top public universities of India by almost all ranking agencies. It is a residential green campus with over 25,000 students, opting 339 program of studies offered by 11 departments, 9 centers, 7 colleges, 2 polytechnics, 3 academies, and 3 off-campuses under 13 faculties. Among these, Faculty of Life Science is a flagship faculty which has been ranked number 1 among the life science faculties of all Indian universities by QS ranking as well as Times Higher Education. We are only behind Indian Institute of Sciences as per these rankings, and this speaks a lot about the standard of research and teaching being conducted by our faculty members. The faculty presently has 6 departments of studies, which include biochemistry, botany, zoology, biotechnology, wildlife science, and museology. The biochemistry department, which is organizing this lecture in memory of one of our illustrious faculty member, has been doing her research since its inception. The teaching of biochemistry in this university started back in 1951 by Professor A.R. Ridwai, one of the papers at master's level, and M.Sc. Biochemistry was started in 1970. Professor Abdul-Majid Siddiqi joined back the Department of Biochemistry in 1962 after completing his PhD from University of California. He was responsible for giving direction to teaching and research of biochemistry at AMU. He, along with his students, Professor Salimuddin and Professor Hadi, and many others, were responsible for creation of full-fledged Department of Biochemistry under the newly established Faculty of Life Sciences in 1986. Center of Wildlife and Ornithology and later departments, Museology, were added to this day. In the meantime, in 1986 itself, a separate Institute of Biotechnology was established as a semi-autonomous unit under the stewardship of Professor Salahuddin, a renowned protein chemist. This has recently been added to the Faculty of Life Sciences. The faculty members in all departments conduct high-end research on both classical and applied aspects on animals, plants, and microbes, right from biodiversity to stem cell research, and have been publishing in top journals of the world, including Nature and Nature Communications. We have research collaboration with a number of countries, including North America, several European countries, Japan, China, Korea, and many others. Our students are working in top research laboratories of the world, and both teacher and staff have received a number of national and international awards and are fellows of academies. I am confident that our faculty members will continue to do high-end research and would follow the footsteps of their mentors. It is with this approach that we remember our teachers who made unparalleled contribution in their area of research and guidance. I once again welcome our guest and the participants, and I hope the lecture presented by a guest speaker will be of great value to all our guests and participants and students. Thank you very much. Over to Sajid.
Thank you, sir.
Thank you.
Thank you, sir, for giving a beautiful introduction of the Faculty of Life Sciences, Department of Biochemistry, and Aligarh Muslim University. A student would definitely cherish their history in the blessed department, faculty, and university. Now I would like to invite Dr. Shahid Jamil, a fine biologist of India, who is the son of our beloved Professor late Abdul-Majid Siddiqi, to enlighten the audience with his valuable thoughts. Presently, Dr. Shahid Jamil is the director at the Triveni School of Biosciences, Ashoka University, Haryana. Please, sir.
Thank you very much, Sajid Saab. And thank you very much, Tabish and Wasim Saab, for organizing this lecture at AMU. And to my younger colleague and friend Anurag for agreeing to give this lecture. Anurag is one of the leading thought leaders in this country at this time, and I'm very proud to have been associated with him and that he agreed to give this lecture today. Let me just spend maybe the next 6-7 minutes talking about who my father was. And if I can just share a slide. You'll have to give me a minute. WebEx is a little different. Okay, I hope you can see the slide.
No, not now.
Not yet. Okay.
पहले स्लाइड को ओपन करके फिर बॉटम में शेयर करना, उसको बस क्लिक कर दिए. उसके बाद
it's coming. It's coming.
Yes,
Now you can see it.
Yes.
Right.
Yes.
All right. So welcome to the memorial lecture. How do I move the slide? Okay. As a scholar, my recollections of my father is about three things. About how honest he was in dealing with people. And there I would, those of you who are old Aligarh hands would know that he had many occasions to take advantage of the situation, but he never took undue advantage of any situation. He was brutally passionate about his work, very honest about his work, and most of the time my meetings with him used to be in his lab. I have also seen him get down on his knees and clean the floor of the lab and clean the gutters of the central instrument facility when it was being built up. So he was extremely passionate about what he did. And the third thing I remember about him is that he was not afraid of new challenges. He learned computers when he was close to retirement. He brought in computer-aided education to the department. Under the International Union of Biochemistry and Molecular Biology, he also had a satellite meeting on computer-aided education in the department, which some of you may be old enough to remember. But after his retirement, he did many other things. For example, he learned how to type in Urdu on a computer and composed a whole book that was left by my grandfather. When he passed away, he was in the process of writing a laboratory manual, a biochemistry laboratory manual, with the help of some undergraduate students in the department. So that's who he was. Some of the academic profile has already been given. He came to AMU after finishing his intermediate at Lucknow Christian College and finished his BSc honors in 52, his M.Sc chemistry in 53, and then was appointed a science teacher in the Unani Medicine Faculty, Tibia College, where he remained till 59, proceeding to University of California at Berkeley to do his PhD. And then the department the same year shifted to University of California, San Francisco, so that's where he finished. He came back to India immediately after finishing his PhD to serve back at his university, first as lecturer in biochemistry, and then rose up to the ranks of becoming professor and created the Department of Biochemistry, of which he became the first chairman in '86, became the first dean the same year of the Faculty of Life Sciences. And in his career, he guided 40 PhD students and 39 MPhil students. His research work was on lipid metabolism and enzymology. In his PhD, he worked with a man named Vic Rodwell, who, by the way, is still active. I met Rodwell two or 3 years back at Purdue University, where he is an emeritus professor. And he's one of the co-authors of Harper's Biochemistry. Some of you may know that book. There he worked on bacterial metabolism of mevalonic acid, which is now, the work is now part of textbooks. His research at AMU led to a US patent for combating high cholesterol. These patent rights were taken by Cal Biochem way back in the early 1970s. The concept that he had given of HMG-CoA reductase inhibitors was really the impetus to the discovery of statins that many of us use today. During the course of his research at AMU, he also discovered two isoflavones from chana, Sicer, and this he showed to have lipid-lowering properties. He also showed the protective effects of HMG on alcohol-induced lipidemia. So his entire research was really directed towards lipid metabolism and the enzymes involved in lipid metabolism. For the benefit of more biochemistry-oriented people, here is how cholesterol is synthesized, starting from acetyl-CoA to HMG-CoA, and really the rate-limiting step is the conversion of HMG-CoA into mevalonate, and this is the step that is catalyzed by the enzyme HMG-CoA reductase, as shown on the right, and he spent a long period of his research working on this enzyme HMG-CoA reductase and finding ways to inhibit this enzyme in the process of inhibiting cholesterol biosynthesis and therefore reducing the lipid load. So that's the molecule HMG, which he showed to be an inhibitor of the enzyme and had lipid-lowering properties. And what I show here is lovastatin, which Merck marketed in 1987 as the first statin molecule. I will simply end by showing you a very old picture that somebody sent me today. This morning, one of his students sent me this picture. And in this picture, you can see him here, always happy among his students and colleagues. Professor Hadi is here, Professor Salimuddin here, Professor Rahed Noor Khan here, Imrana is here, and my very close friend Riaz Mahmood, who's a faculty in the department. Riaz is here. I think this is Nahid, this is Bilqis Appa. So this apparently was a picture taken at the BSc student freshers party, which was a sort of tradition in the department. So I will simply end there, hoping to have given you a flavor for what the man was. Thank you very much.
Thank you, sir, for sharing wonderful memories of your father with us. Now, I would like to invite Chairman Professor Mohammad Tabish for his precious thoughts on today's webinar. Please, sir.
Thank you very much, Dr. Sajid. Honorable Dean of the Faculty of Life Sciences, Professor Wasim Ahmad, our distinguished speaker of the day, Dr. Anurag Agrawal, the Director of the Institute of Genomics and Integrative Biology, New Delhi, Dr. Shai Jamil, Director, Trivedi School of Biosciences, faculty members, and dear participants. Assalamu alaikum and very good morning to all of you. It is indeed an honor for me to welcome you all to Professor Abdul-Majid Siddiqi Memorial Lecture today, titled The Value of Interdisciplinary Research, a Story of Maths, Biology, and Medicine. The lecture aims to tell our young scientists about the interdisciplinary research, which is an essential part of today's research and development. The contributions of and life of Professor Abdul-Majid Siddiqi have already been elaborated by Dr. Shai Jamil Saab. But having the privilege of being his student myself, I would like to share my personal views on him. Professor Siddiqi was more than a teacher for us. He was a guide, guardian, and everything you need at a place far away from home. He gave us a homely feeling at the new place. It was indeed because of his efforts, hundreds of alumni of this department are shining across the world. His efforts helped the department to become one of the first departments in India to teach biochemistry honors at undergraduate level in the year 1986. Today, the department offers courses of BSc, MSc, and PhD. A section of biochemistry is also running at the Women's College for BSc girl students. The department is well funded by different funding agencies and recipient of UGC SAP, DBT Builder, and DST fees, to name a few. Every year, a large number of students qualify national level tests like CSIR JRF, UGC JRF, DBT JRF, UGC NET, etc. The faculties in our department are very dedicated towards research and publish their research articles in high impact journal. In the fond memory of Professor Majid Siddiqi, we are having this memorial lecture by an internationally renowned scientist, the director of Institute of Genomics and Integrative Biology, Dr. Anurag Agarwal. Dr. Agarwal completed his MBBS and MD from All India Institute of Medical Sciences, New Delhi. He did his PhD from Vallabhbhai Patel Chest Institute, University of Delhi. He worked as an assistant professor at Baylor College of Medicine, Houston, USA. Dr. Agarwal, an accomplished researcher of pulmonology and mitochondrial dysfunction, specializes in experimental and computational approaches to understand biological and clinical aspects of respiratory diseases. He is currently working on relations of obesity, asthma, and diabetes, genomics of low lung function of Indians as compared to rest of the world, and use of information technology to improve community health. He has around 200 publications to his name, with several research articles and reviews cited over 2,000 times. His articles have been published in journals of very high repute like Science, The Lancet, etc. He has citations of more than 21,000 in last 5 years alone. Dr. Agarwal has several awards to his name like Lady Tata Young Research Award, Shanti Swarup Bhatnagar Prize, National Bioscience Award for Career Development, to name a few. Dr. Agarwal joined the Institute of Genomics and Integrative Biology in the year 2007 and has been holding the position of director since 2017. I believe his lecture will help us enhance our knowledge about interdisciplinary research. With this brief introduction, I would like to invite and hand over the mic to Dr. Agarwal for the memorial lecture. Thank you.
Thank you very much, Professor Tabish, for a very, very kind introduction. My thanks to all the organizers, my thanks to everybody else who's here, and also to Professor Jamil, whom I have known for many years, for the ultimate honor of inviting me here to give this talk. Without taking much more time, I'm going to start sharing my slides. Let me know once they're visible. Okay. Can you see my slides?
Yeah, you can make it full screen, sir.
Okay, I have made it full screen. On my screen, it is showing full. Let me try. It's not full screen yet for you?
No, I cannot see. Maybe it will take a while.
Okay, let me try sharing. Let me stop this and share it once again.
Yeah, maybe you can do that.
Okay, I've stopped sharing this. Let me try to share it again.
I think, Anurag, you may have to open it from within WebEx.
Oh, open it from within WebEx. Okay.
That's what I just discovered.
Okay. No, that.
I think it's coming. Takes a while.
Let me know if it comes.
Normally what we do simply, yes, it is coming.
It's again not full screen.
If you open your document and then you click at the bottom of WebEx share and then select the file that takes up automatically.
Let me do that. Give me 1 second.
To the right, Anurag.
Yeah, I'm just, I managed to screw something up. Okay. It is a bit unusual. Let me stop sharing everything and let me...
Sir, you open your file on the desktop.
Yeah, that I have done.
And then you go see the bottom share at the bottom. WebEx के bottom में देखिए share एक.
हाँ, वो तो हो गया, वही मैंने किया था.
जैसे आप क्लिक करेंगे तो आ जाएगा ऑटोमेटिकली. फिर आप क्लिक कर लीजिए उस फाइल पर.
या अब आया?
हाँ, आ तो रहा है लेकिन ये साइज छोटा है. पहले जो आपने शुरू में अपलोड किया था just before the in the very beginning it was full screen. हाँ, साइज थोड़ा बड़ा हुआ.
नहीं, go to the view.
Yeah, I'm going to the view.
हाँ, view से भी कर सकते हैं.
Yeah. Let me go to the outline view. No, that's not the view.
No, outline view नहीं.
Yeah, I can make it bigger like this. That is.
Share से भी कर सकते हैं.
हाँ, share तो ये actually यहाँ तो आ गया है.
View.
Here we are.
Presentation.
Present online. Third link.
Present online. नहीं, ये तो नहीं लग रहा. If I do this, ये तो कुछ.
ये कुछ और है.
ये कुछ और है. Weirdest. Maybe I'll do something. I'll start. I'll do this first, and then basically I will. Let me stop. Let me start the slideshow first, and then let me share. Sometimes that works. And
you start the slideshow.
Yeah, that's what I will do. Okay, good. Now let me share this. This might work. Has it worked this time?
No, we're still seeing this.
Still, it is not full screen, I guess.
Tell me what you know. That's screen one. Does this look better?
Yeah.
Okay. All right. So it's a screen sharing system. Okay. Something to learn to new every time one does it. Without much ado, one of the things that intrigued me when I was reading about Professor Siddiqi's life, that his BSc and MSc were in chemistry. It is PhD in biochemistry at a time when probably very few places in India even had biochemistry departments. It made me think about this slide I talk about often. The fact whether the disciplines in science are real or imaginary, something we create because science is ultimately one, and the degree of disagreement we have between scientists regarding the purity of their disciplines. In this particular slide, you will see how mathematicians are at one extreme, believing that their science is possibly the purest. Medicine, you will notice, is not even on the slide. You have chemistry, biology, there is no biochemistry. and you have sociology at one end of the spectrum. And this leads, in my opinion, to some of the biggest problems that we see, which are that, when you teach a subject, that's because it's defined in a particular way, it's very well organized, it's a particular syllabus, it may have a degree. In fact, you can almost define a subject by the fact you have a degree in it. If you don't have a degree in something, is it a subject? This clean, well-defined organization of education is in great contrast to the real world. In a real world, you have problems. These problems can be volatile, uncertain, complex, ambiguous, and trying to evolve the knowledge for providing solutions never follows as clean a path as a subject would indicate. And this is something we all know as disciplinary, multidisciplinary, inter, and trans. It's a slide I like to show often. When people work in their own areas, they're working in disciplines. You put all of them together in a university, you've got multiple disciplines in one place. But until people actually start interacting with each other, you don't get interdisciplinary areas. And then comes a higher level synthesis. They must have been biologists, they must have been chemists. When they work together, you would still have good research coming. But at some point, the disciplines start merging together into something that is transdisciplinary, a higher level synthesis. Now, for me, it's a very important personal perspective. My beginning in science was possibly computer science. That's what I really wanted to do. Along with many other people who were confused regarding what they want to do in life, particularly with a father who was from the medical science, I joined AIMS for MBBS. I continued with medicine for a while, medicine, pulmonary disease, critical care, shifted at some point to experimental research, shifted at a future point to data science and public health. And currently, sometimes I still wonder, what do I do now, apart from doing research, but that's what leadership positions are about. I would like to say that everything I will tell you today is a generalization regarding how one should cross boundaries, how one should not necessarily follow any one particular silo that one gets put into, but this generalization, like any others, is false and perhaps useful to some. One very important reason when you decide about the value of crossing boundaries is why you're doing research. It's a question that everybody needs to answer for themselves. As long as there is a knowledge gap with defined boundaries that you're trying to answer, you're usually comfortable within a subject. But it's a question, larger question, that you're trying to answer. You will never find that your own subject will contain all the answers. In fact, if the answer was that easy to find within your own subject, the likelihood is that many people would have found it already. Most difficult questions of every subject are not answerable within the subject itself, almost always require insight from people outside the field. And of course, another reason to do research is a step in career progression, in which case one chooses a question for an optimal mix of answerability and reward. And there is nothing wrong for any young person who wants to take the third reason to do research, but you will find as your career progresses, you will feel compelled to answer questions that are coming from within, and that's when this problem becomes more and more. This is a slide I show often. A person goes to a drunk person, asking him, "Did you drop the quarter you're looking for over here?" He says, "No, I dropped it 2 blocks down the street." "And why are you looking here?" "Because the light is better here." That is the motivation that I always find is very important while trying to think of merging disciplines. One reason to merge it is you need more light. If you can only search in a limited area where the light of your own area shines, you really cannot find all the missing things. So let me give you a few personal examples and then generalize from there. At this point, I was a medical student. The problem was measuring lung volumes in patients without making them pant against a shutter. And the reason we do that is when you make a person pant against a shutter, Boyle's law comes into operation. Pressure volume swings pretty much is held constant. So delta pressure and delta volume can be connected to each other. And using that, you can solve certain equations. Now, this was a domain in which physiology and biomedical engineering would be the solution domains. So since 1980, it was known that the combination of AV resistance and volume could be measured during quiet breathing, but the volume was requiring a panting maneuver against a closed shutter while sitting in a closed box. The question was, why can't we measure volume of lungs when we measure airway obstruction in quietly breathing people? And if I look back at my career, this was possibly the simplest equation I ever wrote. You know how to measure the product of airway resistance and thoracic gas volume, another way of saying lung volume. If you were to measure it twice, once with the instrument and an additional resistance that you know, you can make this measurement twice. If you make this measurement twice and simply were to rearrange the two equations, you will get the solution of thoracic gas volume. It basically the difference of the two measurements that you made divided by the resistance that you yourself added. And this is a very simple device that you could create. I mean, you don't need to be an engineer to create this device. You would create a couple of shutters. You would create additional resistance. You can change the path of airflow. When shutter number 2 is down, the person will breathe through the additional resistance. When shutter number 2 is up, the person will breathe straight. When shutter number 1 is down, you'll be making the measurement using normal methods when a person is breathing against a shutter and cannot really exhale or inhale anything. This gave me a very nice introduction into the world of science. I must say all my life, was spoiled because this is the paper that when we sent it, it got accepted in the very first time of sending, the Journal of Applied Physiology of the American Physiological Society, with only one comment coming from the reviewers, I wonder why nobody thought of this. And this became a patent and so on. And unfortunately, never in my life has that happened to me again. That, a paper goes in with the reviewer saying, oh my God. And unfortunately, all the next, 30 years of my life have been about rejections and going back and forth, like many of you will share my grief. But I learned something important. There is much scope to improve upon what it is. There are many blind spots between the eyes of different scientific disciplines. I cannot believe for a second that if physicians had gone to people who work with these equations and told them, look, my patient is having trouble breathing against a closed shutter. And the reality is when you do this on a normal person, it's actually very easy. But do you ever want to know the volume of a normal person? A pure physiologist does, but a clinician who's taking care of people with COPD, he does not need to know the volume of normal people. He needs to know the volume of sick people. And sick people do not breathe well against shutters. That simple gap that possibly was not communicated because clinicians were not routinely speaking to biomedical engineers and physiologists left me with a very easy space to enter with my first scientific publication. And 12 years later, I think I was still beginning. I don't think one ever ends. And the problem that we were looking at was that people with obesity, people with hypercholesterolemia, were very high risk for developing asthma. And this was not behaving like normal asthma. It was not responding well to steroids. And we asked our question, could mitochondrial dysfunction, which is well known to be important in obesity and metabolic syndrome, be a connecting link also to lung disease? And the solution remains this time for experimental physiology and cell biology. We liked the idea of focusing on mitochondria. It's a very yin-yang kind of an organelle. It has very critical roles in maintaining cell function, but also very important as a trigger of the innate and adaptive immune system. Very little work was done in lung disease, and little work being done when you have a good reason to believe it might be important is a very good reason to do something. And that's how we went forward. I won't talk to you a lot about our mitochondrial work because a lot of it was classical biology. But at some point, we asked ourselves a very simple question. If mitochondria are so important in the genesis of asthma, could donation of healthy mitochondria reverse the asthmatic phenotype or reverse other lung injury phenotypes? And we were lucky that because of some very good work done by a very excellent student who joined the lab, Dr. Tanveer Ahmed, who's currently at Jamia Millia, we were able to show, and mitochondria here are those green structures, that mesenchymal stem cells can donate mitochondria to epithelial cells. Now, once this was established, and we tested this in so many ways, we could show that if we took MSCs and gave them inside the lungs are very easy organs to work with. They are an external surface of your body, which means if I want to give something to the lung, I already have the nose and the mouth. I can just go through that, inject whatever cells I want, And what you will see is the green little dots coming in the second photograph. And these are donated mitochondria, which are healthy, coming from mesenchymal stem cells. What we were able to show further, we were able to find the motors that move these mitochondria. And this happened to be something called MERO1, which was already well known in neurons, again, something we just borrowed from another field. And we were able to create a variety of cells. Now, this one I'll spend a little bit of time on. MERO high, you can see my cursor. MERO high are the cells that have high levels of MERO and can donate lots of mitochondria. MERO low, right after that, are the ones that cannot donate mitochondria. So now we have created mesenchymal stem cells with healthy mitochondria that either can be donated or cannot be donated. And what you can see is that if you cannot donate mitochondria, then you cannot rescue mitochondrial dysfunction. If you can donate mitochondria, you can rescue mitochondrial dysfunction. Now with this done, we went back to the asthma model that our lab had established for many years. And we did something very simple. So sham is a normal mouse. OVA is a mouse with allergic airway inflammation due to a foreign protein called ovalbumin, which is nothing more than egg white. OVA plus M.Sc MERO CC is simply a control M.Sc. The next one is also a control M.Sc. And what you can see is that the inflammation goes down. When I give MERO high, and inflammation is these little dots surrounding the airway that you see over here. Lots of little dots. When I give MERO high, the entire inflammation just disappears. The lung epithelium looks absolutely healthy. But when I give an M.Sc, which cannot donate mitochondria, the MERO low, the top right, the inflammation does not go away. This proved for the first time the importance of mitochondria unequivocally in the genesis of allergic inflammation and asthma. And while there are a number of scoring parameters in histology that are at the bottom, I will not spend much time on that, but come back to why I'm saying that things become interdisciplinary very fast. And this again is the same thing, except that now look at the brown. Brown is airway remodeling. You can see in C, this is cockroach extract. Cockroach extract leads to severe remodeling of the lung, the airway particularly. You can see MERO high, the dark brown is all gone, but MERO low, the dark brown absolutely remains. And again, when you look at something called airway hyperresponsiveness, you give something to make the airway contract. The mice with MERO high, shown in yellow, are almost like naive mice. They do not react at all. While mice with MERO low, the dashed line, they react a lot. Y bar is resistance. If the airway contracts, the resistance will be very high. But in the process of doing all these experiments, we got these wonderful images. And these are beautiful mitochondria. And what you can see is when you trigger inflammation, here we are using rotenone, a mitochondrial poison, the mitochondria start changing shape. While doing hundreds of experiments, we realized we need to do nothing. We can just look at mitochondrial shape inside lung cells, and we will know whether the mitochondria are healthy or dysfunctional. Now that's nice. That's a nice biologist perspective. You can write it in a few texts, but if you wanted to measure it, you wanted to make a quantitative index of the number of mitochondria that are round. For 24 hours, you can see round mitochondria, and the number of healthy mitochondria that are like threads. Now, how would you do it? You have plenty of students. You could ask them to sit and count. But there are better ways. You could first define the shapes. You can see that we chose 3 words. In retrospect, I think I could have chosen better words, but we chose elongated, blobs, and donuts. We could typically see elongated are the healthy, donuts come in the middle, blobs come in the end. And we asked a chemical engineer who knew no biology, but was visiting the lab. And we asked him, can you write a machine learning system that automatically classifies the mitochondria for us? And this first time, we were using something simple called random forests. I will not bore you with the details, but pretty much they create decision trees, which you can train. You can feed them a number of examples of blob mitochondria, donut mitochondria, and linear mitochondria. And after some time, the decision trees will train themselves to get the right weights of different parameters such that they can classify them. You do not have to put in the parameters. It's random to start with. They're random vectors which train themselves as the algorithm runs. And what you can see here is that he was doing a good job. Now in this picture, you can see automatically the machine is classifying blue as the threads, which are normal. You can see red blobs and you can see pink donuts. And you can see that better in a high-powered view. And now we could go through thousands of images we had collected under a variety of circumstances, with machines doing the classification, machines making the measurement, and machines giving us output like this. As inflammation rises, how do the different shapes change? There were many more figures in this paper. Which one corresponds well to ROS? Which one is reversible? If I were to give an antioxidant, which shapes will revert? Which shapes will not revert? And very quickly, within a few months, we had another paper out, the first machine learning paper out from the lab, on using shape recognition in mitochondria to understand mitochondrial function. And all this came because we were willing to let a chemical engineer come to our lab and figure out how can he add value to our lab. I was not personally capable of any of this, although computer science was one of my passions earlier in life. And machine learning was something that automatically grabbed our attention as the next frontier of medicine. The next 100 years of medicine are entirely going to be about brand new things. It's not going to be so much about just biology. It's not going to be about just the devices. It's going to be about inferences we will draw from the data. The size and complexity of this multidimensional characterization of patients in the future is going to be so difficult for the human brain to handle that we will see very complex diagnostic and prognostic categories, formulas that can simply not be captured in language. You cannot capture what a random forest does in language. It is weights on trees, combination of weights, and you simply cannot describe it other than make sure it works. And this is true for a lot of machine learning. This was another very interesting phase of the lab. I will not speak much about it, except to say that Dr. Pritesh Sethi joined the lab as a medical doctor, working on pulse measurement, Ayurveda style, to build a device. We had hired an engineer. After some time, the engineer decided he wanted to do biology, in which case the Pritesh decided that he wanted to do what the engineer was supposed to do. Dr. Kaundinya Desi Raju came in as a young person who was not allowed to take up maths, because in South India, they do not do maths if they go into medical. But he continued with maths on the side and gave the 12th exam because he felt one day he will do research and the maths will become useful. And both of them showed a passion for getting into data science while also with a background in medicine and doing biological work. And Pritesh worked on time series data, and he is now he must be finishing his India Alliance Fellowship and is a professor at IIIT Delhi. And Kaundinya Desi Raju is also India Alliance Early Career Fellow and is at THSTI. But this strengthened this idea of moving between disciplines in our lab. I'll share some of the works, but not all of what they did with you. One of the works we got involved in was how do you apply machine learning to a very simple question? Can I say that my asthma patient is about to get an exacerbation? Why is this important? We do a wonderful job of controlling asthma today, and we use a lot of drugs to do it. And some of the important drugs we use are inhaled steroids. Now in children, it's a trade-off. Inhaled steroids reduce your height. So does uncontrolled asthma. Between three categories of children: children with uncontrolled asthma, children with controlled asthma but on inhaled steroids, and children with controlled asthma but not on inhaled steroids, you will lose some height with inhaled steroids, but you lose even more height for lack of control. So what you would like to do is, if you can predict the person is likely to exacerbate or not likely to exacerbate, you can modulate the steroids that you give to these patients better. And using a few hundred children followed up every 3 months for 5 years, we are trying to do exactly that. We do lung function testing at every visit along with clinical data. And we find that oscillometry parameters, and this is again a random forest algorithm, this is how you get the output. You basically try to put the various parameters back into the model and see what works. And what you see at the bottom, the ones in the green, are X and R. X means reactance. It's a engineering physics word that describes the ability of a system to store and return energy. you see R, which is resistance, which all of you know. So many of these parameters can be calculated as you put pressure wave oscillations into a lung and measure the feedback and try to calculate the mechanical properties. It requires almost no cooperation, unlike oscillometry. And we are able to determine with almost 80% accuracy which people will exacerbate in the next 3 months. We haven't gone to the next phase yet, which is, can you use this input to reduce and increase inhaled steroids in a way that with minimal amount of steroids given, you get maximal control of asthma. One of the other interesting derivatives from this is, and again, how you end up crossing devices, crossing boundaries all the time, oscillometers are 20 lakhs, and they're all heavy devices kept inside labs. So we realized if we really want to take this technique that we had built, all the AI work that we had done, but we actually want people to benefit from it, it goes to doctors' clinics. Because most healthy asthmatics don't go to hospitals, they go to doctors' clinics. You need to build a portable oscillometer. And that's the other thing that we did along with Cognita Labs in America, a medical grade oscillometer approved by the FDA in 2020, which works on batteries and is very small, the amount of size that you can hold in your hand, suitable for children, suitable for clinics. So again, if your problem is not just trying to understand what happens in asthma, but to ensure that your understanding actually translates to clinical benefit, there is no option except to work with AI people at one end, medical doctors at one end, physiologists at one end, and device makers at one end. And a very similar problem came recently in AI when we were trying to figure out how to use X-rays to determine whether somebody has COVID or not. All very challenging problems, all very important, but this simply involved cross-connectivity. This involved cross-connectivity in the sense that you need to know what you want to do and connect to the right people to do it and encourage young people, which I would say is the most important thing to pursue these directions. So moving on to a larger, even bigger question. How do we improve respiratory health of Indians? The previous one was easy. The previous one was how do we measure something? How do we measure it more effectively? How do we measure it in a way that every doctor can use it? Now we want to have true understanding. Now we're getting closer to biology. And the solution domains become even more complex. Beyond biology and medicine, this is public health. There's a lot of modeling is required, a lot of molecular work is required. And if I had to choose one line to characterize it, I would say such a question is typically classified as biting off more than one can chew, and it's typical of such questions. To start with, we did something simple, which is pure technology. We set up multiple sites for healthcare measurements around the country, which were basically health centers built inside shipping containers. The entire data was on the cloud. We could collect a lot of data from around the country directly from the point of caregiving to integration on the cloud. We were able to use it because of the support of Hewlett-Packard at multiple sites around the country. We saw over 1 million patients by 2019. We understood that why people come to see doctors in terms of respiratory complaints being very, very high, but most of them were little non-specific. The problem with this approach, it gave us some insights, was that the data was usually of poor quality. And while you can try to do lots of things with data to make even poor quality data a little bit useful, but at the end of the day, you have to realize that doing lots of data crunching on fundamentally low quality data has limited value. So we went on to even larger, better design study. In this study called the Poseidon study, what we did is we asked 10,000 doctors from around the nation, and this is the distribution around the nation. We asked them to let us know what on one day did all their patients come with. They had a questionnaire. Every patient was a column. Every possible thing organized by organ systems the patient could have was a row. And the doctor simply had to check off little things on that sheet. And those sheets were collected with the help of CIPLA from all over the country. And using this data on 200,000 patients seen by 8,000 doctors around the nation, we're able to come up with some insights like this. And I have not shown you the full broken down details, but you could see that at every decade of life, the biggest reason for people to come and see doctors were respiratory problems. If you were to take circulatory problems, so basically this is called an alluvial graph. Again, something very interesting that came out of our interface between medicine and maths. Alluvial graphs are fundamentally network analysis, something very commonly done in mathematics, something very commonly done in informatics, but almost never done in medicine, at least in 2015 for public health. And we could basically build networks of different types of complaints people had from around the country and see how they play out across decades of life. So whatever is the biggest node sinks to the bottom. So respiratory is the biggest at every part of life. Circulatory, you can see, becomes most important after around the age of 50. You can see endocrine is really unimportant. That's mostly diabetes if you break it down. Becomes really important across India in the 40s, late 30s and 40s, and then almost merges with the circulatory, giving you a huge number of people who have both cardiovascular disease and diabetes. You can look at when eye problems occur, you can see many things. One very interesting thing you will see is anemia and female genitalia, which is basically gynecological problems. And you can see that they're both very separate. In the 2nd to 5th decade of life, they come together and then they separate out again. That is classical menstrual anemia, a very obvious public health problem in India. And if you were to split our data by cities and towns, you could see cities do not have it, but our towns and villages still do. And this is something that you can do with large data collected in a reasonably clean way. Our first effort was a very unclean way. Our second effort was a slightly more clean way, but not research grade data collection. And so we could find that small towns had higher respiratory symptom frequencies than big cities. And the question that we had to our mind is, why is that? The big cities are the one with the bad pollution. An obvious answer comes to mind, it could be because of dirty cooking fuel. And the government is clearly launching, that programs to create better LPG penetration. But where is the support that this is because of dirty cooking fuel? And since we had collected the data simply from doctors' practices, there was no way for us to know whether those people whose data we had were using dirty fuel or clean fuel in their houses. And this is the other advantage of big data. You can connect things in a way that normal research cannot. So for 1 lakh subjects we could find, the government had already published local LPG cylinder sales in census data. We could geotag the doctor. We could make an assumption that a patient who comes to see a doctor comes from near the doctor. It's not a perfect assumption. It's a reasonable assumption. For that geographical area, we can calculate a new variable. That variable will be the sales of LPG cylinders per unit per population. Now you can imagine if this variable shows a low value, it is likely that people in that area are using dirty fuel. If this number is a high value, it is likely that people in that area are using clean fuel. Now that's another assumption, but that's what data science and medicine really intersect. Putting these two things together, now you can build what is called a Bayesian map. It's also called directional graphing for those of you who are mathematically inclined. It basically says if A, B, and C are correlated, if I can hold B constant while varying A and C does not move, and I move B and C moves, then A acts on C via B. And if you have sufficient number of people, and we had 1 lakh people in this, you can permute the data in enough number of ways that you can come up with quantitative associations between variables as well as estimates of direct connectivity. And the most important thing I will show you is this yellow and this green. The green is the LPG cylinder sales per unit population. The yellow is the prevalence of obstructive airway disease as per the doctors. And what I want to show you is, if I were to change the value of LPG cylinder per unit population from 14% to 85%, the number of people with obstructive airway disease as per the model, it's a model, will drop by half, which means half the chronic obstructive pulmonary disease of this country would come down simply by making clean fuel available. And that is something, of course, that was appreciated. Very interestingly, as a side story, we had to do this analysis for Lancet Global Health to accept our paper. But after they saw the analysis, they accepted our paper, they told us to remove the analysis. And we asked them why. because they said the level of evidence is not high enough for it to be a conclusive part. Say they would not like to publish it, but showing this made them comfortable that the possibilities of the work that we had done were high. So one of the most interesting comments I ever received in my life. So moving from there, some other things that we saw by some other data that was coming was that spirometry, which basically takes how much air you can forcefully exhale after maximum inhalation, it totally how much air can you blow out after you breathe all the way in, was quite low in Indians. Now this was troublesome. This was troublesome because John Hutchinson, who invented the spirometer, called vital capacity because he found it was a measure of vitality. The more air you can blow out, the longer you live. The less air you can blow out, the shorter you live. And obviously it has to be adjusted for height and some other factors. But by and large, multiple studies from around the world have found this. Why is this troublesome? For the same height, for the same gender, for the same age, for the same weight, for the same socioeconomic status, South Asians can blow out the least air. So FVC is the total amount of air you can blow out. 0 here stands for a white European or American would be able to blow out. FEV1 is how much air you can blow out in 1 second. And what you can see is South Asians can blow out only 70%, 30% less, 1/3 less air than a white American would be able to blow out. And remember, I have already adjusted for age, gender, height, weight, everything in these kind of comparisons. Does it then mean that Indians are high risk for dying of cardiac disease? Because that's what Framingham study showed is very likely. People who blow out less air, in fact, even 25% less, have double the risk of premature death from cardiovascular disease. Now, if that is true, then should we be calling this normal? If you take any Indian equation for measuring lung function, it will say this is normal. This brings us to the question of, at one end, people like me are getting worried about the fact Indians are not blowing out enough air on all these measurements we are making. We are seeing that exposure to dirty fuel, many other factors possibly may be driving these problems across the country. But yet the poor lung function of India is called normal in our equation. So the word normal, the norm, the normal, and the normative are 3 very different words. Norm simply means common versus uncommon. It's often confused with normal in the medical sense, which means healthy versus unhealthy. And normative actually is supposed to mean desired standards. And when these 3 words get conflated, confusion starts. So what we did is something very simple. We took about 1,200 subjects. The ones in red are from Vellore. The ones in blue are from the National Capital Region. We measured their spirometry parameters. You can see here forced vital capacity, the total air you can blow out, as well as the ratio of what you can blow out in 1 second. And we plotted them by age as a Z-score. Why as a Z-score? We simply don't care what is normal. We are simply trying to say on a global distribution where that person would be. So a Z of -1 would mean the person is at -1 standard deviation from the median of the world. -2 means -2 standard deviation from the median of the world. And what you can see is that Indians, even at a younger age, are starting low, around -1.2. And by the age of 50, they're down to -2.2. That's really low. At -2 standard deviation, you are at the bottom 2.5 percentile of the world. Any small size lung that gets progressively smaller, if you want to use that word as an analogy, cannot be a healthy thing. If you were to look at the bottom right, the difference between what our lung volume should be versus what it is observed to be, you can see we are almost 1 liter short by the time we are 20, and we are more than a liter short by the time we are older. Now this is troublesome. This is troublesome because this could mean that we have the beginning of airway disease subclinically even before we realize. So we went back to those oscillometers that we built, and that's where, doing multiple things in a lab really became very useful to us. And this was work by Mohit Agarwal, shown here on the left. All the work I'm about to show is basically led by him. And what you have are measurements of small airway disease. If the y-axis is high, small airway disease is likely. If the y-axis is low, small airway disease is unlikely. And what you can see, and these are all healthy people. These are all self-declared healthy people with no symptoms. As your ZFC, FVC becomes negative, which basically means as your lungs are smaller, the likelihood of your having, and these values are not likelihood by themselves. By the time you cross 0.2 on the top right, you have about an 80% chance of having small airway disease. You can see even after -1Z, your risks of having small airway disease are tremendous. These people are between the age of 30 and 50. And what you can see from here is people with small vital capacity, which represents about 1/3 to 40% of the Indian population, are already showing very sensitive oscillometric markers of small airway disease in their lungs. This is not something that we can say with 100% confidence because these are ultimately physiological markers. We don't actually have sections of their lungs, but I would tell you this with a reasonably high degree of confidence, maybe at least 80%. The next question then you would ask is how early does it start? And the only way you will get the answer is to go to children. So we did a study across India in a cohort called SOLID. You can see the expansion over there. Children from 6th grade to 12th grade, pretty much covering all over India. Jawahar Navodaya Vidyalaya, which pretty much as all of you know, is a group of boarding schools catering primarily to the lower plus lower middle class. Perhaps we do not see affluent India over there, but I would argue to you that we see real India over there. So 2,611 kids, 1,400 males, 1,200 females from 14 different sites in the country, spanning almost everything possible you can imagine regarding pollution, hills, valleys, urbanization, and so on. I don't have the time to share all the data with you today, but I'll show you some important things. I'll show you the summary of two things. One is the vast majority of our apparently healthy children have inflammation. That can be seen on the bottom right. And there is a clear phenotype. WHTR is waist to height ratio. What you can see is children in cluster 2, which is, and cluster 1 and cluster 2 are both around 35% of the people. Cluster 2 are people with waists that are good relative to their height. Nobody is obese. This is simply in the normal zone. Our average BMI is in a range of 24. There is no question of obesity in this cohort. These people have a higher ZFVC. They have lower levels of inflammation, lower levels of interferon, lower levels of interferon TNF alpha, lower IL-8, lower IL-17. On the other side, you have kids who are apparently healthy. They have low ZFVC. They are thin. They are very thin. They're not emaciated. These boarding schools give them adequate nutrition, but they have very high levels of inflammatory cytokines. They have high IL-17, IL-8, TNF alpha, interferon gamma. What could this be? What could make a child thin, unable to gain weight despite adequate caloric intake? These are not very poor kids, and stunted in the way their organs and their body is developing. And the answer is unknown to me, but I suspect the way I have phrased it would make many of you think of the same thing, chronic gut infections. And exactly in line with such suppositions, we found that smaller rural places were more likely to have people belonging to cluster 1 compared to places like Delhi and the outskirts of Delhi and big cities, which had more people belonging to cluster 2, which is the healthiest cluster. So basically telling us that early in life, the availability of clean fuel, the availability of clean food, the availability of clean water are possibly setting the shape for the way India will progress as a healthy nation. Nothing very surprising, I would say. We did a huge number of studies. I mentioned perhaps a huge amount of India Alliance money. I got a senior fellowship from the India Alliance to do this. But I think we definitively show here where we need to change as a country if we want a healthier next generation. So this is a simple explanation. The reason people die early is because of the susceptibility factors all linked to each other. Chronic low-grade inflammation, poor development of organs, poor development of arteries, all starting in early life, chronic inflammation, poor quality air, poor quality diet, giving you multiple things that become interlinked to each other in countries. These include diabetes, heart disease, and respiratory disease. I'm going to start winding up now. I'm aware of the time. And I'm going to come back to the mitochondria. You will see that I become very interested in microbes. I continue to believe that mitochondria are very important as well. And so what we are really working on right now is a lot of this Indian respiratory dysfunction that is also marked by mitochondrial dysfunction and change in the microbes in our gut all connected. Prashant Bajpayee did this work. He's finishing his PhD now. And he found that diseases linked to gut microbiome have a very significant overlap, almost 2/3, with diseases that have previously been linked to mitochondria. So out of 114 diseases on PubMed, currently linked to gut microbiome, currently means actually 2 years, 80 of them had previously been linked to mitochondria. This was encouraging. How could they be linked? Microbes make a huge number of metabolites from your gut. Mitochondria are like cousins of ancestral microbes that have gone and lived inside your cells. Would these metabolites be doing things to change mitochondrial health? And again, I come back to mathematics and informatics, pull the data, annotate, overlap, path enrichment, all standard informatic work. A little bit more than that, build flux balance analysis, create entire metabolic maps of mitochondria. When we started, there were 400 reactions. Today, our lab has put 1,800 reactions. We are still sending out the paper. After you do all this, feed in metabolite by metabolite, try to define an objective function for such as ATP production, ROS generation, and then go to wet biology. Test these metabolites. See if they're actually doing what we expect them to be able to do. And I'll give you just one data to show, yes, we are. So one of the things we are using here is mitochondrial shape. I told you that earlier. In extensive data from our lab in respiratory cells, just shape analysis tells us a huge amount about mitochondrial health. So we trigger using a molecule called TMOG, bad mitochondrial health. You can see the little swollen things, swollen mitochondria under TMOG. We are artificially inducing mitochondrial dysfunction. And we are giving metabolites that our metabolic models come from bacteria in the gut, but seem to be capable of changing mitochondrial health. And you can see we are restoring it. The shapes are again going back to normal. So brown was normal vehicle. And you can see after giving DMOG, the shapes changed. These are linearity. And then they go back after giving ornithine. And there are many other things we have done, including seahorse. But again, the wet biology and the dry biology running side by side. Of course, you have to keep reading. You can do all the work that you want, but you have to keep identifying what other people do as well. So pyrroloquinone quinone is a molecule that is already approved for human use, is already there in your diet, influences diabetes, is described to be a mitochondrial vitamin. Nobody knows how it works, but we tested it in our, and we cannot figure out how to place it in our models, but it seems to work. So we tested it in our wet lab. It worked very well, and we took it to human trials. So this will be the first example of a microbe-derived mitochondrial targeted therapeutic being tried for respiratory disease. We started the trial in 2019 for COPD and lung fibrosis at AIIMS in Delhi. Unfortunately, because of COVID, all respiratory trials that involve breathing into machines ended. So we only have 3 months data. In fibrosis, it made no difference. But in our limited number of people for whom we had 3-month data for St. George Respiratory Questionnaire, a symptomatic questionnaire of COPD, in people receiving standard therapy versus standard plus PQQ, you can already see significant differences and improvement in the way they function. So with that, I come full circle, starting my biological career in IGIB from pure biology, moving towards informatic machine learning things like shape analysis, moving towards purely informatic things like trying to figure out what metabolites bacteria might be producing that might be working on mitochondria, going back to human roots, trying to do something that I believe will change respiratory health because it will cater to one of the most susceptible organs in our body, the mitochondria, which bad air, bad food, bad water, all seem to be giving us bad health. I'll stop here. I think I've merged enough number of streams together to confuse everybody, but I hope I was able to give you a flavor of how to bring various things together. Thank you very much.
Thank you, sir, for your thoughtful lecture, which will definitely inspire our students and show them a path to achieve the success in career as well in social and the personal life. Now I would like to invite Professor Riaz Mahmood for a vote of thanks. Please, sir.
Riaz, we cannot hear you. Maybe you just want to plug or unplug your earphone and speak into the computer.
भाई अजीम माइक को जरा देख लीजिए, माइक ईयरफोन के निकाल के लगा दीजिए अजीम।
Tabish, are you planning to have any questions?
From the attendees, they have been muted for technical reason, and that is done from the computer center.
Oh, I see.
I wish I could, but
Yeah. Since I'm not muted, may I ask a question?
sure. I mean, all the panelists can ask
Okay.
question. I was
Wonderful.
talking about yes.
Yeah. So, Anurag, thank you very much. It was really wonderful. I've listened to bits and pieces of this, but I've never really heard the whole story together. So it was really wonderful. And I think a few things come to my mind. One, as you said, how the best answers come from interdisciplinary work. And you have demonstrated that very nicely in your talk. The second thing is, how science can lead to public policy. And this is something you demonstrated with your work on cooking fuels and drawing that correlation. And thirdly, what impressed me most
Hello.
is, yes, we can hear you. The third thing which impressed me most is while most people who do microbiome studies do association and only correlation studies, you are actually beginning to share causality. And that's very, very important. So just one technical question, Anurag. When you were looking at the different types of mitochondria, don't the blobs appear to be sometimes elongated because of the plane of view that you're looking at?
Oh, absolutely. So basically, the size matters. So when you look at it, so a long cylinder cut cross-sectionally can look like a dot. It will not look like a blob. And the blobs are actually almost spheroids, ovoids actually.
Okay.
So no matter which dimension you cut them in, they'll still look like blobs.
Right.
And the dots we don't count. So that's something again
Okay.
that the ML system learns, the size.
Oh, thank you. Okay. Riyaz, over to you.
Can you hear me now?
Yes, yes.
Yes, sir. Yes.
Okay, finally. Okay, I'm sorry about this. Dear Professor Anurag Agarwal, Professor Wasim Ahmed, Dr. Shahid Jamil, Professor Tabish, and Dr. Sajid, my colleagues, friends, and all participants of this lecture, it's my privilege to propose a vote of thanks on the occasion of the second Professor Abdul-Majid Memorial Lecture. This lecture is held in memory of the person who laid the foundations of biochemistry in AMU. First and foremost, I, on behalf of the entire Department of Biochemistry, would like to thank Dr. Anurag Agarwal for delivering a really interesting and almost fascinating lecture. I hope everybody has benefited from it. As Professor Tabish mentioned earlier, Dr. Agarwal is former Shanti Swarup Bhatnagar Awardee in medical sciences and is currently the director of the Institute of Genomics and Integrative Biology, New Delhi. We are indeed fortunate to have such an eminent scientist deliver this lecture. Thank you, Dr. Agarwal, for taking time
Thank
out
you. Thank you.
from your busy schedule to give this really wonderful lecture. We are really grateful to you for taking part in this lecture. We are also thankful to Dr. Shahid Jamil for helping us in arranging this lecture. Dr. Shahid Jamil is no stranger to our department. He's a frequent visitor to our department and has given us several lectures here. He's also a Bhatnagar Award recipient and a virologist of repute. Dr. Shahid has helped us in numerous ways, right from the initial to the final stages, most importantly by liaising with Dr. Agarwal on our behalf. And I think it will not be wrong to say that it is due in large part to his efforts. This lecture has been held today with Dr. Anurag Agarwal as the speaker. Thanks are also due to Professor Tabish, the chairperson of our department. He did most of the planning and execution of those plans from our end. Dr. Shahid and Dr. Tabish were in constant touch for some time to work out the modalities of arranging this lecture in our department. The idea of this memorial lecture was conceived by former students of Professor Majid Siddiqi. They also fund 2 scholarships for B.Sc and M.Sc students of our department in Majid Saab's name, and I would like to thank all of them, and especially Professor Aftab Ansari at National Institute of Health, USA, who took a personal and keen interest in ensuring that the lecture was held. Thanks are also due to members of the Computer Center who arranged the modalities of this lecture, Professor Wasim Ahmed, Dean Faculty of Life Sciences, and the University Administration for cooperating with us all the way. This was the second Professor Majid Siddiqi lecture. The first one was held 2 years back. For some reasons beyond our control, it could not be held last year. But we hope that this lecture is held annually in the future, and we continue to get as eminent scientists as Dr. Agarwal to deliver these lectures in the future. Finally, I would like to thank Professor Agarwal again and all those who have joined this lecture. Thank you so much.
Thank you, sir, for the wonderful vote of thanks. Now, in the last, I would request Professor Mohammad Tabish to utter the final remarks about the today's prestigious Professor Abdul-Majid Siddiqi Memorial Talk. Please, sir.
Well, I would like to express my sincere gratitude and appreciation to all my colleagues and participants for attending this lecture. Lastly, I would like to thank again Dr. Anurag Agarwal for wonderful lecture, Professor Wasim Ahmed, Dean Faculty of Life Sciences, Dr. Shahid Jamil, Professor Riaz Mahmood, and Dr. Sajid Khan for making this event successful. Thank you once again and have a good day.
Thank you very much, sir. Goodbye for today.
Thank you, Anurag. Bye-bye.
Thank you. Bye-bye.
Bye-bye.